Yes. I'm sharing my screen. Can you see my screen? Just let me know.
No, ma'am.
Yes, now?
Yes, ma'am.
Okay.
Yes, ma'am.
Okay, fine. So we were discussing the chapter. We already started the chapter absorption by root. We'll just discuss it freshly again. Yesterday we discussed a little very much. So now we will discuss it again. Now you all know absorption by roots. Huh. Absorption by roots. So in this chapter, we are going to study absorption by roots and what are the processes involved, like how roots absorb water, what are the different processes, and where is absorption needed. Okay, need for absorption and in which all the processes it is needed. So you all know in plants, roots absorb water. You have studied this in class 6, I suppose. So roots absorb water. Roots are mainly responsible for absorbing water and mineral nutrients, and they conduct them to different body parts of the plant, like leaves, flowers, fruits, stem, etc. So let us read this need of water and minerals for plants. Now, besides being a constituent of protoplasm, water is needed inside the plant body for four purposes. You all know the four purposes why water is required inside a plant body: photosynthesis, transpiration, transportation, and mechanical stiffness. Just a minute. Yes. So photosynthesis, you all know why in photosynthesis water is needed, because the main constituent is water. Carbon dioxide plus water gives out glucose and oxygen. Oxygen is released out, glucose is stored as a food. Then in transpiration, large quantity of water gets out of the leaves. Okay, sometimes the plants absorb more water and the water is supposed to be released out from the pores of the leaves. So in transpiration also, water is required because it has to be thrown out or released out from the leaves, which gives a cooling effect to the plant. You all know transpiration have some factors also. Some of the advantages of transpirations is, okay, there are two main advantages. One is it gives cooling effect, and one is it increases the cell sap content. Okay, it increases the cell sap, or we can say the content of the cell inside the plant body. So that's why in transpiration water is required. Then third is transportation. That means water has a good flow. So that's why the minerals and nutrients which are absorbed by the soil, by the roots, absorb the minerals and nutrients from the soil. So these nutrients are transported with the help of water. Okay, they cannot transport alone. They are transported along the water molecules from different parts of the plant body. Then mechanical stiffness. Fourth one is mechanical stiffness. Why? Mechanical stiffness means getting turgidity or the rigidity, means getting swollen. Okay. Water provides turgidity. That means it sometimes water when in more condition, when the water is more inside the plant, the plant will just swell up. So it will give it mechanical support or mechanical stiffness. Okay, the plant tissues will get stiffness inside them because of the swollen nature. Because the water is inside is more, so the plant or the tissue will swell up, and thus it will help in mechanical stiffness. You got this much? Is it clear?
Yes, ma'am.
Yes. Then, yes. Then we have some paragraph on mineral nutrients. So basically, you all know nutrients are present in the form of salts and ions, right? Salts such as nitrates, phosphates, sulfates, and ions such as calcium, potassium, chlorine, etc. So these nutrients, or we can say the salts and ions, why they are required inside a plant body? What is the purpose? So that these elements help in synthesizing the variety of compounds within the cell. Okay, there are so many compounds within cell. So these salts and ions, the nutrients, collectively known as the nutrients, they help in synthesizing some of the compounds in the cell. Underline this, the last third sentence. These elements are required as constituents of cell and cell organelles as well as in the synthesis of a variety of compounds or enzymes within the cell. Underline that much. Then come to page number 38. Now we are actually starting the chapter. Just now we discussed that where the water is required and what all purposes it is required, why it is required and all. Now the main chapter starts from here. Characteristics of roots for absorbing water. Now you all know roots are responsible for absorbing water. So they should have some characteristics also. Without any characteristics, they cannot easily absorb water. So what are the characteristics? So there are basically three characteristics. First is they have huge surface area. Second is they have more amount of cell sap, greater cell sap is there. And third one is they have thin walls. Now, let us describe the first one, huge surface area. Now you all know root have root hairs in them. Okay. Not only the root, who absorbs the water inside the root? The root hairs. Okay. One root consists of hundreds of root hairs. You all know this thing you have studied in your class 6 standard. So one root, you can see one root is present. So suppose this is a tap root. Okay. This tap root will have many fibers. Okay, small thread-like fibers it will have. So these fibers are more in number. I am making only 5 to 6 in number, but these are actually more in number. These are actually goes above 100 or up to 100. They are in number. So there are many small fiber, thin fiber-like structures known as root hairs. These are known as root hairs. So they are large in number. They are hundreds in number. So because of their number, they occupy a large space. Like you can see in this diagram, they are more in number. They are more than 100. Sometimes they are 100 in number. It can be less also, but approximately 100. So they occupy large space in this particular root area. Okay, large space is occupied. Because of that space, the surface area is increased. Okay, like I don't know if I've given you the example of cotton or not. Like if we take a small piece of cotton, it can only absorb little amount of water. But if we take a big piece of cotton, like a big gauze piece, if we take, it can absorb more water because it has more surface area, right? It is large in number. Similarly, these root hairs, they increase the surface area of the root and thus they are able to absorb the water from the soil. Clear? So, when carefully examined, each rootlet would show 100 of root hairs. If all the root hairs of this plant were to be laid end to end, they would cover a length of many kilometers. You can see a freshly germinated seed showing the single root with root hairs. You can see this root, main root, it is a tap root and many thin fiber-like structures are arising from this. These are the root hairs. Clear? So these root hairs increase the surface area of the main root and thus it is responsible for taking up or absorbing the water from the soil. So this is very easy. Yes, whose mic is on? Yes. Now the second is greater cell sap. The second characteristics why root can absorb water is the cell sap. Root hairs contain cell sap of a higher concentration than that of the surrounding water. That means, you know, just a minute. This is very easy to understand. You can read this paragraph and it is very easy. But I am still, making you understand with some diagrams so that it will be better for you to understand. So you know, root is in the soil. Now you know soil has water molecules. Okay, moist soil is there. We plant seeds where soil is moist. So water is more and soil is more. This is water. Okay, water and soil is there. So this root here contain more water content. That means more fluid is inside this root here as compared to the surrounding soil. Like this soil has water, like it has water, but this root here has more fluid content inside it. Now what is that fluid content? What is a cell sap? It is all the fluid containing the inorganic salts and the waste materials which is coming out from the plant body, the food and some different compounds, some nitrogenous compounds. All these together make the cell sap. You can write if you want cell sap. Just a minute. You all know what is a cell sap. If you want to remember it quickly, you can write it. Cell sap means cell sap is a fluid. Okay, it is a fluid which contains what all? The inorganic salts, inorganic, organic, salt, and plus waste material. The waste material which is coming out from the plant body is also included in this fluid. Waste material. Then what all? Plus, I told you food. The food which is made in the plant body, inside the leaves. This also contributes in the fluid, the cell sap. That means all the content in this particular plant, all the contents in the plant, the inorganic salts, waste material, food, and the nitrogenous compounds. Nitrogenous compounds. All these things are present inside the plant body. Okay, everything we can see in the plant body, and this is the whole content of the plant body. So this whole content is known as cell sap. This whole content, including all these inorganic substances, salts, waste material, food, and nitrogenous compounds, includes the or contributes to the cell sap. So all these things are more. These things, these four things, the other things, the fluid, extra water is more inside the root. Inside the root, it is more. Okay. And just regarding the surrounding, as compared to the surrounding, as compared to the soil in which the root is present, the fluid content in root is more. So that's why one characteristic is this, that the root has more amount of cell sap in this. Root hairs are the extensions of the outer epidermal cells of the root. They also contain large vacuoles filled with a solution of cell sap. Some salts are dissolved in it, and the cell sap, therefore, usually has a concentration higher than that of the surrounding water. This characteristic is an important requirement to draw in water from the outside, that is for the occurrence of osmosis, which is described later. So, for the purpose of osmosis, this characteristic is very important. Now, you all know what is osmosis. Just shortly, I will say osmosis is the movement of water molecules along with the minerals from higher concentration to lower concentration through a membrane. Through which membrane? A semi-permeable membrane. So, in the process of osmosis, this character of high cell sap is very much applicable. Clear? So this is the second point, greater cell sap or high amount of cell sap. First one was large surface area. Second is high amount of cell sap. Then the last one, the third one is the presence of thin walls. Root hairs have thin walls. Now you all know plant cells have two types of walls. Animal cells have only one type. Which one is that? The cell membrane in animal cell. But in plant cell, two types are there. Cell membrane is there and outer to cell membrane, there is a cell wall also. This is a cell membrane. So outer to that is a cell wall also. Cell wall is also present in plants. So That means the cell wall is very thin. Okay, these walls, this cell membrane, the cell wall, these are very thin. Now the cell wall is thinner than the cell membrane. So what it will do, the cell wall will allow some substances to enter into the body. Okay, this is very thin. This is a semi-permeable membrane. It is very permeable. So it will allow the substances, water molecules and dissolved substances to move freely in and out of the cell. That's why root hairs have thin walls. That's why they will allow some substances which will eventually lead to the main characteristic for absorption of the roots because of these thin cell walls. Clear? Is this much clear?
Yes, ma'am.
Yes, ma'am.
Okay. Okay. So these thin cell walls, it will allow some substances, the water molecules, dissolved substances to enter into this body. So that's why absorption will be more because of the presence of this thin wall. Now the next, okay, just come to this diagram of a root hair. Just have a look at this. Now look at your book on page number 38. You have a diagram, figure 4.2. A single full grown root hair and two more in successive stages of growth from the epidermal cells. Now this is the enlarged, or you can say an anatomy of a root hair. Root hair looks like this. So this main, you can see this big, this, just a minute, yeah. This straight big thing is a root. This is a root, and this extension, you can see an hair-like extension. This is the root hair. So you see the labelings: epidermal cell wall, cell membrane is there, cytoplasm is there, vacuole, nucleus is there. So root hairs beginning to grow out. Now you can see this extension here. When you consider this whole root, you can see some extensions, small extensions like this. So these are the root hairs which are beginning to grow, and this is a fully grown root hair. Okay, so you can see root hair consists of what all? Root hair has a nucleus, has a vacuole, has a cytoplasm, has a cell membrane, has a cell wall, etc. Okay, just look at your book. This diagram is very important. It may come in your exam. So just look at it carefully. Look at the labelings carefully and try to by heart this diagram. So I'm not making the full diagram because the whole diagram is given in your book. So just consider this diagram. It's very easy. Just try to make it in your notebook once. You will get it. Now we have 4.4, absorption and conduction of water and minerals. Absorption and conduction of water and minerals. Yes. So the entire mechanism of absorbing water and minerals from the soil by the roots, its movement through the thickness of the root and subsequently its upward conduction through the stem is the result of 5 main phenomena. That means the movement of water, that means the process of absorption is the result of 5 basic phenomena. Okay, this absorption is not very easy. You will think that, okay, roots are present, hairs are there. So these root hairs will directly absorb water. No, there are 5 phenomena acting here, 5 processes acting here, which further gives the result of absorption, this absorption characteristic of root. What are the 5 main phenomena? Imbibition, diffusion, osmosis, active transport, and turgidity and flaccidity. Now, first we will discuss imbibition. Now, imbibition is a slightly new word for you. You have discussed, you have known what is diffusion is. In 6th standard, you had diffusion, osmosis, active transport. Turgidity also you must have known. Flaccidity is little new, and imbibition is a newer topic. So what is imbibition? It is nothing but the phenomenon by which the living or dead plant cells absorb water by the surface attraction. That means some dead plants which, who are not able to attract water or absorb water, they do surface attraction. Like seed, if we consider a seed, you can see a seed is coated, it is shiny, so it cannot usually attract or absorb water. It cannot absorb water easily because it has coating, it is hard, rigid. Okay, so this seed, there's some these seeds which are not able to absorb water directly. They absorb through surface attraction. Okay, so this process is known as imbibition, where the particular living or dead plant cell, anything, it could be living or dead plant cell, absorb water by surface attraction. By the surface, they absorb their water. So, substances which are made up of cellulose or proteins are hydrophilic. They imbibe water or moisture and swell up. Example, dry seeds. Now, as I told you the example of dry seeds, you know how dry seeds, like if we put a seed in the water, they will take time to absorb the water and they will eventually swell up after one day or after some time. If you see the seeds, they will swell up. So they are basically doing what? They are absorbing the water through surface attraction because they don't have any permeable membrane to absorb water. They do not have thin wall, so they will do surface attraction. And the examples you can see: dry seeds, wooden doors, wooden doors, if water is put on doors, they will swell up on contact with water on exposure to moist air. Due to imbibitional pressure, seed coat ruptures in case of germinating seeds. It is also an important force in the ascent of sap. Now, due to this force of imbibition of water, like when surface attraction, water is attracted by the surface, absorbed by the surface, then this seed coat ruptures, and then further the water enters the seed and it swells up later. Eventually, it swells up. So this is the same in the case of ascent of sap. You all know what is ascent of sap. This was also in your 6th standard. Increase in the sap content or increase in the cell content. How it will increase? Eventually, when the water is getting inside, other fluids are also present inside, so the cell content will increase. That increase is known as ascent of sap. Now, this was imbibition. Is this much clear?
Yes, ma'am.
Yes?
Yes, ma'am.
Yes. Okay. So second, we have diffusion. You all know very well what is diffusion. It is the movement of water molecules from higher concentration to an area of lower concentration. Very easy. This definition you can see: free movement of molecules of a substance, solvent gas or liquid from the region of the higher concentration to the region of lower concentration when the two are in direct contact. That means they should be in direct contact for the process of diffusion. The water molecule should be in direct contact. There should not be any membrane between them. Like in osmosis, we have semi-permeable membrane. So here that's why it is mentioned in bold letters, direct contact. They do not have any membrane in between during diffusion. So what is diffusion? The movement of water molecules from the region of higher concentration to the region of lower concentration when they are in direct contact. Okay. So there's an example given of diffusion. Place a sugar cube or a small tablet of a soluble dye or a crystal of potassium permanganate in a beaker containing water in one corner. The sugar, the dye, or the potassium permanganate slowly dissolves and spreads in the liquid. Eventually, the molecules of the substance are diffused or disturbed uniformly throughout the water. You can see if we place a crystal or a cube of sugar, so it will eventually start dissolving in the water. In the movement of molecules, there was no obstacle of any kind in the path. Now, you know, diffusion, we are not using any obstacle or we are not using any membrane. Okay, direct contact is there. So, of any kind in the path of the molecules of the dye, they could move freely in water in all the directions. So as we have told earlier, diffusion does not require any membrane. We are not keeping any obstacle or any membrane in between the liquid. It is just a direct contact between the liquid and the solute particle which we have put in the liquid. Maybe crystal or a sugar or a tablet, anything. So direct contact is there. You can see the diagram A, B, C. In A, it is placed in a corner. In B, it is eventually dissolving in the water. And C, you can see the whole water turns into red color. Okay. So you can experiment this. You can take some colored substance and you can experiment this. The whole water will turn into colored substance. Let us see how the definition of diffusion applies in this case. The molecules of the dye are more crowded or more concentrated in and near the tablet of the dye. These molecules move away farther and farther in the regions where they are fewer or absent. This movement continues until the molecules are uniformly distributed. Stirring with a spoon or glass rod hastens the process of uniform distribution and you get a homogeneous solution much faster. So this is what we said right now. These molecules, they just try to or tend to distribute themselves in the water molecule because they are not having any obstacle between them. And when we stir it with a spoon or glass rod, it will just hasten up the process. Hasten means speed up the process and we will get a homogeneous solution very fast. So this is the example of diffusion. Then we have osmosis. Osmosis also, you know it very well. The process of osmosis, just a minute. Yes. So what is osmosis? This is very much similar to diffusion, but not that similar because it has some different. Only the first line is similar to diffusion. That is, osmosis is the movement of water molecules from the region of higher concentration to a region of lower concentration. But here, slight difference is there. From a region of higher concentration, that is dilute solution or with a lower solute concentration, to a region of lower concentration, that is concentrated solution or with a higher solute concentration through a semi-permeable membrane. That means movement of water molecules from a region of higher concentration to a lower concentration through a semi-permeable membrane. This is a semi-permeable membrane, SPM. Semi-permeable membrane. So through a semi-permeable membrane, water molecules move from higher concentration to the region of lower concentration. But here, a slight difference is there. What is the difference? Semi-permeable membrane. And also, this higher concentration is in the place where the solution is dilute. And this lower concentration is where the solution is concentrated. Okay. Dilute means the solution has been diluted with the help of water. More water is there. And here the solution is concentrated. That means particles flow from here to here. And you can see this acts like this semi-permeable membrane acts like a chutney, right? We use chutney like when we make tea, we use chutney so that we can, we can take the two different things in different saucers. Like we are taking the tea into the cup and the tea leaves just collect into that chutney and the chutney is acting like a semi-permeable membrane there. That is not properly a semi-permeable membrane, but we are just taking the example to understand this. So here, this tea, when we talk about this tea, the tea leaves are on the chutney, are collected in the chutney, and the tea which we have collected, all the, the tea leaves extract will go into the tea and we will get a concentrated solution here. Right, this tea leaves will make that more concentrated. So similarly here also, this water molecules or the molecules will pass through the semi-permeable membrane and here the concentration will be lower but a concentrated solution. Clear?
Yes, ma'am.
Yes. So let us read this paragraph. An important thing to note in the above definition is that only the water molecules move from the higher concentration to their lower concentration in stronger solution or just a solution. I told you in tea also, while making tea also, only the water molecules, the water, the milk is moving outside, not the tea leaves. So here also the water molecules, because this is a semi-permeable membrane, this is not a freely permeable membrane. Okay, this is not a freely permeable membrane. That's why only the water molecules is passing, and the bigger molecules, the solutes, are remaining here itself. So this was osmosis. So osmosis is slightly different from diffusion in the way, because it has a semi-permeable membrane. Also, the higher concentration is a dilute solution, and the lower concentration will be a concentrated solution. Now, page number 40, come to page number 40. With reference to a cell, osmosis can be either inward or outward, depending on the extent of concentration of the solution surrounding it. Endosmosis, exosmosis. There are two types, endosmosis and exosmosis. Now, endo means inward and osmo means push. Exo means outward. So, with reference to the cell, they are differentiated into two, outward and inward. Now, what is endosmosis? Is the endo means inward. So, endosmosis is the inward diffusion of water through a semi-permeable membrane when the surrounding solution is less concentrated. This tends to swell up the cell. Like, for example, what can we give as an example of endosmosis? By reading this, what endosmosis example will be obviously a seed, a seed which is getting water from outside. That means from outside, seed is absorbing the water, the diffusion, or we can say the inward movement of water through a semi-permeable membrane. The seed coat is acting as semi-permeable membrane, and the water from outside is getting inside the seed. Okay, and it will, what is written in the last? This tends to swell up the cell. So this is in the case of seed. Okay, it will swell up the cell. Or we can also take one more example. We have resins, kismis. Okay, we have kismis. So kismis also, the cell wall is very thin. This is the semi-permeable membrane is there. So it will absorb water from outside. When we place kismis in some liquid like water or rum, anything, so it will swell up. It will absorb that particular liquid and it will swell up because of the semi-permeable membrane. So we can take the example of kismis is a very perfect example because it is having a semi-permeable membrane. So we can give this example of kismis in endosmosis, where the water molecules from outside is absorbed by that particular thing which is placed in the water. So here the example is kismis. Resins you can write in English. And then exosmosis means the outward diffusion of water through a semi-permeable membrane. When the surrounding solution is more concentrated, this tends to cause shrinkage of the cell. Now, we have taken kismis as the example of endosmosis. We can take in exosmosis, we can take the seeds also. Why? When we when the seeds inside the water, when we take them out, and when they are not in the water, and when they are kept for some time outside, like for 1 or 2 days, what will happen? They will start shrinking. That means the water is going out of the cell. Okay, outward movement of water through the cell. So that will tend to cause shrinkage of the cell. So shrinkage of the cell means we can take the example of seed. When the seed is put outside the water for 1 or 2 days, it will start shrinking because it will start losing water. Clear?
Yes, ma'am.
Yes. Okay. So you can see this experiment. Yes. Experiment explaining osmosis. Take some concentrated sugar solution in a thistle funnel. Cover the mouth of the thistle funnel with a cellophane paper or egg membrane or animal bladder and tie it securely. Invert the thistle funnel in a beaker containing water and suspend it as shown in figure 4.4. Mark the level of the sugar solution on the stem of the thistle funnel. This is the experimental setup. As a control, take another thistle funnel with plain water filled in it and suspend it in another beaker also containing water. Again mark the level on its stem. After a few hours, the level of the sugar solution in the thistle funnel in the experimental setup will rise. The level of the water in the thistle funnel in the control will remain unchanged. The level of water in the beaker in the experiment will drop slightly, while the one in the beaker in the control will remain unchanged. Okay. If the water in the beaker in the experiment setup is tasted, it is not found sweet. So we have taken we have taken cellophane paper has been taken, a beaker is there, and we are taking a thistle funnel. Okay, this is an apparatus used in chemistry labs, a thistle funnel. So when we are placing a thistle funnel inside a beaker containing water, and the beaker containing water is having sugar solution. So when this thistle funnel will absorb this water, this is okay. We are placing a cellophane paper in the bottom. The cellophane paper is acting as a semi-permeable membrane. Here the cellophane paper is the semi-permeable membrane. So through this, the water will eventually move inside the thistle funnel, right? You can see in the experiment from the beaker, it is moving inside the thistle funnel through this cellophane paper. So only here we have taken sugar. So sugar will get dissolved in water. It will not be in a molecular form or substantial form. So it will get dissolved in water, and this dissolved waters of, or we can say the dissolved solution of sugar will be absorbed by the cellophane paper, or we can say the semi-permeable membrane, and it will start entering the thistle funnel. So here we can see when this water is entering the thistle funnel, what is happening? The water in the beaker is lowering. Okay, the water in the beaker is lowering there, and the water inside the thistle funnel is increasing by the passage of time. Because of the semi-permeable membrane, it is increasing, it is absorbing all the water, and the water in the thistle funnel is increasing by the passage of time. So this is an experiment shown for the purpose of osmosis. Now you know water inside will increase and the water outside will decrease. Clear?
Yes, ma'am.
Yes. Ma'am. Okay. So osmosis, we have done osmosis, and osmosis is also over, exosmosis is over. Now we have three main conclusions from this experiment. What is the conclusion? In this setup, some water of the beaker has passed through the cellophane paper to enter the thistle funnel containing the sugar solution. Sugar from the thistle funnel has not passed into the beaker. The cellophane paper has acted as selectively or differentially permeable membrane. It has allowed water molecules to pass through, but not the sugar molecules. So we have also taken sugar molecules inside the thistle funnel. Now sugar molecules being big molecules, they will not pass from the semi-permeable membrane or the cellophane paper. So that's why in this experiment, it is shown what is the conclusion. The beaker, the water inside the beaker has been absorbed by the cellophane paper and entered inside the thistle funnel. But the sugar molecule which was already present inside the thistle funnel is not coming out, or it is not distributed in the water inside the beaker. Why? Because sugar molecules being large in size, they cannot pass through this semi-permeable membrane or this cellophane paper. That's it. So this was the conclusion from this experiment. Now we have modified version of experiment 2, but the time is over. So we will stop here. We will discuss the next topics in the coming classes. Do you have any doubt?
No, ma'am.
No, ma'am.
Okay.
Ma'am, from third chapter, what you have to write in copy?
Yes, third chapter.
What do you have to write in copy?
Third chapter is genetics, right?
Yes, ma'am.
Yes, so in genetics, you have to write the short answer type and the descriptive type and the skill type E, C, D, and E. You know, you do not have to write A and B. Okay.
Okay, ma'am.
Clear? Yes.
No.
So you can, during exam, you can learn from the notebook itself, and you have to maintain the notebook. I told you yesterday also, maintain the notebooks. There are 10 marks for notebooks. Clear?
Ma'am, where
Yes,
do I
ma'am.
submit the bio?
Syllabus of the for exam.
Yes, what happened?
Ma'am, syllabus for exam.
Syllabus for exam, I'll give you in next. You have to study the second chapter, chromosomes, the third chapter, genetics, and I'll think of giving cell because cell you have already learned in 9th standard. Yes, cell was already there in the 9th standard. Okay, take cell also. Cell first, second, and third chapter. Clear? For FA1.
Yes, ma'am.
Ma'am,
Yes, Rohan?
ma'am, where do I submit that bio chapters 2 and 3? Chapter 1 I have submitted, but in Moodle there is no that assignment
Yes, I
option
have
for.
not I have not given an option of submitting the assignment. So maybe father will ask to bring your books one day to the school. So we are thinking of like calling you to the school and just checking the notebooks in hand. You don't have to submit in the Moodle. Okay, so we are thinking I'll tell you like what father will tell us. So I'll inform you. Is it clear?
Yes, ma'am.
Yes, ma'am.
Okay, ma'am.
So you just have to prepare, you just have to write all this in your notebook, the descriptive type, the short answer, and the E1, the skill type questions. Do this much in your notebook and maintain the notebook. That's it.
Okay,
Yeah?
ma'am.
Okay. Okay, then bye everyone.
Bye, ma'am. Bye, ma'am. Thank you.